Alright, this is exercise video number 11, and for my exercise today, I ran 1 mile with a pace of 9 minutes and 10 seconds. And so what was happening during this exercise is, in order for my muscles to contract and then the antagonist muscles to relax, signals had to be sent from my afferent neurons into my brain to be processed and then out through efferent neurons in order to direct my muscle with what to do. And so whenever these action potentials were sent to my motor neurons and then eventually sent into the motor unit where they were acting upon, the signal was received by the dendrites and then integrated by the cell body and then traveled down the axon in a propagating manner, so just depolarizing, repolarizing, over and over again, and eventually it went into the axon terminal. And that depolarization caused the synaptic vesicles to fuse with the membrane and exocytose neurotransmitters right near my muscle cells, right near the receptors on my muscle cells in the synaptic cleft. And so these neurotransmitters either were EPSPs, IPSPs, or no change happened. And so EPSP is contraction. And so whenever my muscles were contracting during my run, EPSPs were sent and binded to my muscle cell receptors, and then that signal traveled along the cell membrane and then down into the T-tubule, and then that signal was detected by the electrical sensing feet, and then that told the sarcoplasmic reticulum to release calcium, and that calcium went and binded to troponin and told trypamyosin to move out of the way so that myosin could grab actin. And so in order for my muscle to shorten, to contract, myosin was able to grab actin once calcium binded to troponin and moved trypamyosin out of the way. And then during that contraction, ADP and inorganic phosphate were released. And so the myosin pulled on the actin and then was able to recock whenever ATP binded again, but couldn't grab onto that actin until calcium was released again from the sarcoplasmic reticulum in order for my muscles to contract.